In DNA replication, the enzyme helicase cuts through and unwinds the DNA, and the enzyme topoisomerase follows ahead of the helicase and prepares the DNA to be cut. And it follows ahead of the replication fork. Single-stranded binding proteins are attached to the separated segments of DNA and they kind of stabilize it. And then the leading strand moves in the 5 to 3 direction. And DNA polymerase 3 comes and attaches on, and it replicates the DNA and adds on the nitrogenous bases. And once the base is added, it goes to just 1 phosphate instead of 3. And then the rest of the bases are added as DNA polymerase moves down in the 5 to 3 direction, and it replicates continuously. And that's the leading strand. And after the leading strand, the lagging strand is also replicated, going from the 5 to 3 direction the other way.
And the primase is the enzyme that goes along the lagging strand, and it adds the primer and divides up the strand for the next step. And then the mRNA comes in. And the DNA polymerase 3 will come in through and fill the gaps with the triphosphates.
And it'll go from primer to primer, so there'll be a gap between the Okazaki fragments.
And replication will occur from the 5 to 3 direction, obviously. So that's why the lagging strand must be replicated in fragments, going the opposite way. And then DNA polymerase 1 will come in and replace the primer with nitrogen bases. And then the ligase is an enzyme that will come in and bond the new nitrogenous bases with the Okazaki fragments all together.
So that it'll be one continuous piece instead of fragments.